Assalamu alaikum. In this video, we are going to talk about blood hemostasis. Basically, hemostasis is a mechanism that leads to cessation of bleeding from a damaged blood vessel. So, basically, this is a mechanism in which if your blood vessel has been ruptured, like कोई cut लगा है या कुछ भी और bleeding start हो गई है, तो the hemostasis will function to stop the bleeding. So, the goal is to stop the bleeding, कि bleeding रुक जाए. And in order to fully function hemostasis, this process has three steps: primary hemostasis, secondary hemostasis, fibrinolysis, and then the tissue repair will start to happen. And these processes basically have the components as proteins, vascular endothelium, and mainly platelets. So let's see each step. First step is primary hemostasis. Basically, it is a step in which there is transient vasoconstriction and platelet aggregation is seen to form a platelet plug. Basically, what happens that if your blood vessel is being damaged, as you can see in image number 1, so your blood vessel is damaged and endothelial cells are being separated. I hope you all know that a blood vessel is made up of the layers of the blood vessels is made up of like first you have basement membrane, then you have a layer of collagen, and above that you have your endothelial cells, smooth endothelial cells. So basically you over here this blood vessel has endothelial cells. uh, um, this is what we study in histology. I hope you know that. So blood vessel is damaged, and because of that endothelial cells are separated and underlying collagen is being exposed. Now the first thing the body will do immediately to stop the bleeding is the vasoconstriction, narrowing the lumen so that the blood over here bleeds less. So you can see over even in this picture there is this curve, or even in this picture you can see the area that is normal, the lumen is broad, and the area that is damaged, the lumen is short. So first thing that happens is vasoconstriction, narrowing of the lumen in an attempt to stop the bleeding. The next thing that happens is platelet appear and they start to adhere to the lumen, to the subendothelial matrix, collagen. To all of this area, the platelets start to aggregate and adhere, like they start to attach, जुड़ना शुरू हो जाएंगे, clump बनाएंगे, and this happens because of various factors that trigger these platelets aggregation. And what are these factors? The first thing that is initial adhesion occurs via pseudopods that platelet form by changing their shape. So what happens that you can see normally the platelets have smooth surface, but as injury happens at that site, platelets form pseudopods. आप देख सकते हैं कि इनके जैसा knob type appearance हो गई है, knob सी appear हो रही हैं. So basically you have a smooth platelet, and then this platelet develops pseudopods. So these pseudopods will help platelets stick together. So initial adhesion is because of that. Second thing, the collagen, and there is this fibronectin that is being secreted by the endothelial cells. These two things will also cause stickiness in the platelets. The third thing is von Willebrand factor, a very important factor that is present in the platelet granules. So basically, these platelets have granules inside them, and these granules are being secreted at the site of injury. And these granules have many factors that we will study in detail in hematology, but right now one of the most important factor for hemostasis is von Willebrand factor. This will be secreted and it will cause the platelets to stick together, adhere. So this sticking is will form primary platelet plug that is called as primary hemostasis. So your primary platelet plug is formed, आप ये देख सकते हैं, this is called as primary platelet plug. So in this plug there are only platelets and there are no fibrin strands. So what happens that if your cut is very small, this process still here is sufficient. That is only primary hemostasis, that is two things. Number one is vasoconstriction and number two is platelet plug, primary platelet plug are sufficient enough to stop the bleeding. And because if the bleeding stops, body will stop the process over here. But if your cut is big, the process primary platelet plug will move further to secondary hemostasis. What is secondary hemostasis? Basically, in secondary hemostasis, you have coagulation to form meshwork of fibrin, and fibrin meshwork will strengthen the platelet plug. So basically, after primary platelet plug is formed, the coagulation processes will start, and these coagulation processes will basically the goal of this process is to form the fibrin. Fibrin will attach with the inside that like platelet plug, and it will tighten the प्लेटल प्लग। इसकी मिसाल ऐसे ही है जैसे आप कुछ सामान लें और उसको आप इकट्ठे रखें, तो ठीक है, इट्स गुड कि वो एक दूसरे के साथ आपने अरेंज करके इकट्ठा रख दिया, लेकिन उसी सामान को आप इर्द गिर्द से रस्सी से भी बांध दें तो वो ज़्यादा अच्छा टाइटली पैक सामान हो गया। जैसे आप एक लोडिंग वाली गाड़ी में सामान रखा है, बट उस लोडिंग वाली गाड़ी को सामान रखने के बाद आप रस्सी से भी बांध दें सारा, तो सामान गिरता नहीं है। सो फिब्रिन इज लाइक दैट। You can see this picture that these are the strands of the fibrin. So if the cut is big, fibrin is necessary to stop the bleeding. So fibrin is basically formed by 3 cascades, and they are very important. The number one is intrinsic, then extrinsic, and then there is a common. And in order to function or these pathways, you need, or your body needs, proteins. Basically, proteins are factor 1 to 13. Then your body needs calcium. Calcium is basically required for each and every step of these pathways, and there is vitamin K that is required for only formation of prothrombin, protein C and S, and a few factors that are needed in this process. So these are basically components that are required in to fully function these 3 steps. You can see how over here steps. The first one is starts from here. The factors will that are involved in intrinsic cascade, then you have an extrinsic cascade, and they will end up in forming 10A component, 10A factor, and then you have a common pathway. In common pathway, you should it's okay if you don't remember intrinsic or extrinsic pathway, but you should remember your common pathway. That is very important. So in common pathway, you have basically prothrombin that is converted to thrombin by factor this 10A factor. And then what happens that this thrombin will convert fibrinogen into fibrin, and this fibrin will form a stable clot. So you should remember this last step. These are very important, and we will see in last slide why these are important. From this whole process, your fibrin meshwork will form, and that will strengthen the platelet plug. So basically, in hemostasis, 3 main things have happened. Number 1 was vasoconstriction, number 2 was primary platelet plug, and number 3rd was this fibrin meshwork, that is secondary platelet plug. So you should remember that these 3 main things have happened till now. Then the next step we have is thrombolysis or fibrinolysis. Okay, in this process, the fibrin platelet aggregate is dissolved after healing. So your secondary platelet plug has been formed, or your fibrin platelet plug has formed, and it's now like a few days or some time has been gone, that the area is now fully covered and no bleeding has happened. So now it's time for that platelet aggregate to dissolve because healing has occurred. So you can rename or define it as like clot retraction and fibrinolysis process. So what happens, you have seen this is a common pathway. Prothrombin will form into thrombin because of factor 10A. Thrombin will form fibrinogen convert fibrinogen into fibrin. So till up to here, you know this fibrin was the factor that was forming that fibrin meshwork. Now you have to stop, your body has to stop this, otherwise this fibrin will keep on forming, and now we don't need that. So what will happen, that there are 2 things, that you have plasminogen that will convert into plasmin into your body by TPA or urokinase, like that process will happen, and this plasmin will basically convert fibrin into fibrin degradation products, and this fibrin degradation products is removed by your macrophages. So what will happen that this fibrin meshwork is now being dissolved, and then your damaged blood vessels will start to repair by connective tissue, and endothelial cell growth will renew at that place, and your whole area will get back to your normal appearance in your body. Then we have the last thing left is coagulation inhibitor. We have heard there are many coagulation inhibitors that are being given to the patients that have bleeding issues, but we will talk just one over here, and other we will be discussed in coagulation panel tests in biochemistry series. There is an antithrombin 3 that basically acts on this. This antithrombin 3 is basically formed by liver in your body, and this will stop the formation of conversion of fibrinogen into fibrin, and then the clots or DIC, that is a very common disease in which different clots form into your body. It is being controlled by antithrombin 3 in such a way, and this is a very famous drug that helps deal with many of the blood issues. So this was a very important topic, as many of your hemostatic processes and blood coagulation panel like biochemistry or blood reports are being interpreted by the factors that we have studied in this. Next we will start the biochemistry panel, and then we will talk things in terms of lab reports over there and interpret things over there. So guys, thank you so much for watching, and if you like the video, do subscribe and share the channel. Thank you so much.